Good morning, good afternoon, or good evening, everyone, depending on where you are in the world. Welcome to the second EMBARK webinar. Today we will have Heike Schmitt coming on to talk about her research, and we will also have our own Etienne Ruppé talking a little bit about his research within the EMBARK program. So the EMBARK program is a program that which is aiming to establish a baseline for antibiotic resistance in different environments, which could be used for monitoring purposes. And during the webinar today, we wish that you keep your microphone muted, and you can type your questions in the Q&A box, which should be available to you. Note also that this event will be recorded and that you can use the hashtag EMBARK2021 on Twitter if you're live tweeting from this in some way. We also have some common webinars. Today we have Heike Schmitt. In three weeks there will be the Morris, and in 6 weeks on the last of June we will have Kimberly Klein. So you're very welcome to those as well, and there should be registration links in the chat box to those. And with that, I would like to introduce our first speaker today, which is Heike Schmitt. Heike Schmitt is the principal investigator at the National Institute for Public Health and the Environment, RIVM, in the Netherlands, and she's also at the University of Utrecht. She works with the role in the environment for the transmission of resistant bacteria and antibiotic resistance genes, particularly the role of the aquatic, farm, and soil environments. But today she will talk a little bit about how the WHO Tricycle protocol can be put into use in monitoring. Welcome, Heike.
Should work.
Yes.
I can see my screen. I'm thrilled to be here presenting to an unknown crowd of attendants in an easy way. So I hope we have lots of international participants here, which is great to be able to host a meeting to many people across the world, because this talk is uniquely devoted to international activities we run at the National Institute for Public Health and the Environment in the Netherlands. So you won't hear anything about this small country in Europe. I start right away, and I guess I don't need to introduce antimicrobial resistance here at all. The only thing I'm going to mention is that AMR is international and multifactorial, and as we have learned from COVID, it can spread across countries in a very short time frame. And I think this is very clearly expressed also by the World Health Assembly, by recognizing that there's a need for a coherent, comprehensive, and integrated approach at global, regional, and national levels in a One Health approach. So this is what this talk is devoted to. It's showing how to bring surveillance, AMR surveillance, into practice in a One Health approach, which is not that easy because you have to be able to compromise. That's basically the giveaway of my talk. Go forward and compromise, because that's your only chance to work together between sectors. But first, we start out with a short overview of existing WHO policies, because this was a WHO activity. So, of course, many of you will know the global action plans on antimicrobial resistance that are called for by the World Health Organization, which include 5 points, from raising awareness, doing research, as we all like to do, working on sanitation, hygiene, and infection prevention, to usage and sustainable investments. One important piece of WHO guidance is on human AMR surveillance within the GLASS system, the Global AMR Surveillance System. This is devoted mainly to clinical isolates, bacterial isolates that present themselves during people having infections and being analyzed microbiologically. This system is directed at a variety of bacteria species which are relevant from a clinical viewpoint. And what is remarkable for the GLASS system is how well the data collection and also the reporting is organized globally. So there's an increasing number of countries participating in GLASS, and they write excellent reports about the status of resistance in these clinically relevant species. So I think that was a blueprint. As often as we see in AMR One Health activities start out in the human sector and then slowly work their way into different sectors. And this is why GLASS is largely advanced. But also on the animal side, WHO has been working intensively also together with other organizations, with the OIE and the FAO as well. One particular document I would like to mention is the guidance on integrated surveillance of AMR in foodborne bacteria. And that includes aspects that are similar to the human surveillance system, in that it monitors resistance in relevant species, but also antimicrobial usage in animals. And also here, countries are moving towards a fully integrated analysis and reporting. Last but not least, and very close to my heart, is also water, sanitation and health, WASH. Because even though water, sanitation and health is even mentioned in the goals for the global action plans, there is still a misconnection between activities in the sanitation sector and the AMR sector. And I think this is largely due to history, due to that the WASH sector consists of environmental microbiologists, engineers, water safety practitioners, while AMR is often geared from the human health sector. So these sectors are not used to collaborate, again, pointing to the relevance of approaching different sectors. But notably, AMR, if you look at the movement of AMR, it moves exactly along the same lines as fecal pathogens that are excreted with human and animal excrements. And also, I think it's important to note that WASH and AMR are integrated also in another way, because if we improve sanitation and hygiene in a given country, most likely this will also reduce infections, and the reduction of infections in general will also reduce the amount of antimicrobials needed. So in turn, we probably will see in the long run also reduction in AMR. But back to Tricycle. I would like to introduce you a bit to the history of Tricycle, how it came about, and also what kind of thinking is behind it and how it changed during the setup. So the Tricycle project is an initiative of the AGESA group. That's the advisory group on integrated surveillance, which was largely devoted at animal surveillance for a long time. And some members of the AGESA group, Awa Idara Kane from WHO, Antoine Andremont, Mark Sopsy and Morgan Scott from Texas, published a concept note which actually led to the Tricycle protocol development, and that started out in 2015. So we are also now some years into the development. And the overall goal of the Tricycle project is to provide member states with a common, simplified and integrated multi-sectorial surveillance system to detect and estimate the prevalence of one indicator with a specific resistance, namely only ESBL producing E. coli. Why that? What are these principles and how do they compare to glass? Well, first of all, the overarching principle here, which we shouldn't forget, is simplicity. Therefore, the idea was to start out only with one indicator, only focus on one bacterium as a starting point, and this was chosen to be ESBL, E. coli, because it has to be simple to be feasible in as many countries as possible, with as little resources as possible regarding both costs, but also importantly, laboratory resources and also training needs. Also, the data management has to be feasible, and therefore it was good to be able to link that up with existing WHO features. And of course, it has to be standardized, as all larger surveillance systems have to be. And often we are faced with the question, why ESBL? Why ESBL producing E. coli? Of course, this is arguable and up to discussion, but the idea was, first of all, ESBLs confer resistance to critically important antimicrobials as defined by the WHO. And then importantly, there's also variation in their prevalence and the colonization rates across continents, but also in time. So we see a change in the prevalence. So it's therefore an interesting parameter to monitor. And very, very importantly, of course, it doesn't make any sense to work on a One Health indicator if the indicator is not present in animals or in the environment. And for ESBL, it was known that that ESBL can be widely found in animals, but also with different prevalences in different types of animals and across countries. And it has also been found in the environment widely. And last but not least, it has also been shown that once countries or farming sectors take interventions, for example, reduce the usage of specific types of antimicrobials, then often we have also found a decrease in the occurrence rates. So we also know that interventions make sense. It's not a type of resistance that is present since the 1940s and is ubiquitous. So we still can do something about it. So that also makes it a relevant parameter. And also to mention here, again, this is only a starting point. Countries are open and free to choose additional indicators as they wish. The Tricycle is a true multi-sectoral collaboration between the human sector, food chain, and the environment. And that means that if you think about surveillance, it's essential that you have a very good body consisting of the management, coordination, and leadership. So we saw during the piloting the Tricycle protocol that collaboration of the sectors is not an easy thing to bring about, as many of you will also know from your countries, probably, because people that don't know each other, institutions that don't know each other, have to get in contact and have to set up collaboration frameworks. And this requires time, this requires management skills, this requires a good knowledge of what is present in a given country. But this is also a crucial outcome of any implementation of the Tricycle, because if you bring actors together on one given project for the first time, it's much more likely that they will also be able to find each other in future on other issues and other criteria or other questions. So I think this is one of the central features. And this is highlighted in the project structure, not so beautifully here, but you see that the true analytical work packages, surveillance in humans, food chain, and environment are only a part of the project. There's also a very important management aspect here from the country, but also up to the global level. WHO headquarters are often still involved. Epidemiology design and analysis should be involved in establishing all the surveillance sampling events. And there's also a link with GLASS, with the other WHO reporting systems and possible extensions. One thing we spent a large time on when we developed the protocol is to ensure that the protocol is as similar as can be between the sectors. And that's actually a large stepping stone, because being an environmental microbiologist as well, I can be thrilled by metagenomic analysis and by qPCR arrays and by many things. And E. coli is just one single bacterium, but we have to realize that other arms, the human and the animal arm, are much less used to these approaches. And so we had to find something that is feasible in all the sectors and that is easy to implement. And that can be done with methods that already exist and are similar to what countries already implemented. So we try to come up with at least standardized culture media and standardized downstream analysis for confirmation of the ESBL we found. And we see that even that was at times difficult to standardize a protocol, because just to give an example, McConkey is the medium in many countries for detection of E. coli in human feces, often also in animals. There are many labs that are well acquainted with these methods, but simply McConkey doesn't work in water. You find all kinds of organisms, but you don't find E. coli, or you find a lot of things that are different from E. coli. So even here, from my viewpoint, we had to argue for 1 year that we just can't use McConkey in water samples until we got all the other sectors convinced that no, we have to sacrifice protocol similarity in order to be specific enough and to be simple enough for countries to interpret what they are finding on their media. So that shows how you have to compromise. To shortly walk you through the aspects of the WHO Tricycle protocol as it stands now, Human surveillance is conducted at 2 different levels, which I find greatly interesting because one of them is similar to the glass system at hospital level, where bloodstream isolates are kept, tested for E. coli, and among the E. coli isolates, the proportion of ESBL E. coli strains is determined. And again, there it's maybe interesting to know that from a European perspective, there was a very natural choice. But from other countries, we often hear that E. coli is really not their major problem. They are much more worried about Acinetobacter. So you have to compromise not only between sectors, but you also have to compromise between countries on the relevance of given indicators. And what I find interesting is that the Tricycle protocol is a protocol where resistance is also studied at the community level, which is still quite rare to find in the open literature. And this is brought about through testing healthy women, pregnant women, when they present themselves as at a general practitioner or for delivery. And I think this will render highly interesting data on community carriage that could be compared between countries. Animal surveillance, where one discussion there was which animal to choose. Poultry turned out to be the easiest one because it's frequently consumed. It's raised around the world. There are limited religious aversions against usage of consumption of poultry. Samples are readily available. A chicken is not as expensive as a cow is. And sampling can be combined with sampling of other bacteria that might be sampled already in integrated surveillance, like Salmonella or Campylobacter. Also, again, the animal friends had to compromise because often animal testing is done through selective enrichment. But here the compromise was to keep it as similar to the human protocol as possible. So the agreement was to have a direct plating on antibiotic spike media, so selective direct plating. Then focus on the environmental arm, because this is what EMBARK is about and what probably is most interesting to you. Well, how to set up a limited environmental sampling system. Yeah, you know you're going to miss so much. So the basic idea is sources of resistance. If you look at resistance in terms of ESBL, which often will originate from animals and humans. So the sources are often animals and humans, not so much resistance that is already present in the environment. And exposure to ESBL present in the environment will often take place through water, for example, through drinking water, but also if surface water is used for irrigation of produce, or if there's direct contact with water through recreation or through other water uses. So the basic idea of the environment testing in the Tricycle protocol was to sample both sources, so human wastewater and animal wastewater, but also sample one compartment that is somehow representative of human exposure or the environment itself, and that is a river. So how is that brought about? How is that implemented? In a city that is chosen within the participating countries, or in 2 cities, there are 4 different sampling locations, 2 of them represented for the sources. So within the city, this could either be a treatment plant or it could be a larger sewage canal serving a larger population with known fecal discharge, and animal wastewater. And again, that could be slaughterhouse, but in many instances, this was a wet market where animals are slaughtered. Then the receiving environment is represented through a river, the river that happens to run through the city. And so one sample is taken upstream, which is not a background sample because this can often be already contaminated, and one is taken downstream. So this is a representation of how the city with an intense large population influences the river concentrations. There are suggestions for extensions if countries are interested. These concern hospital wastewater, but also drinking water. Drinking water internationally is not relevant for all countries. Therefore, we didn't want to include it in all in the protocol itself, because I think it would make little use in, for example, the Netherlands to advocate for ESBL testing in drinking water. But in other countries, this is very well interesting. Sampling frequency for the environment is 8 to 12 sampling rounds spread across a year. in these 4 locations in 2 cities per country. And that means in the 1-year application of the Tricycle project, which is typical, this would result in some 64 to 96 samples. This sounds quite small compared to the human and animal samples. These could be 240 sometimes. But this is also because the environmental analysis is actually the most demanding of all, as I will show now. Well, for the human and the animal branch, what is done is a prevalence study. So presence, absence of ESBL in human and animal feces. But at least from the experience we have in the Netherlands, well, even in the Netherlands, we can find ESBL E. coli in 100% of all raw wastewater samples we are testing. And it would make a great point to study different countries, which probably most of them have 100% prevalence of ESBL E. coli in wastewater. Well, you don't have any distinguishing number as outcome. So what we suggested is that we need the concentrations of the ESBL in water to be able to differentiate between countries. But again, as we all know, concentrations of bacteria in water can greatly vary depending on circumstances as rainfall, seasons. So a mere concentration measurement doesn't also make sense. So therefore, we don't only need to measure ESBL, but our idea was to standardize it, and to standardize it in a very simple way through measuring also E. coli. And by doing that, we can determine the ratio between ESBL producing E. coli and total E. coli. And that bears the advantage that E. coli are often already included in activities at country level on water quality. So they know how to conduct that analysis. To show what countries then can expect as a hypothetical outcome, on the left side, this could be the concentration of ESBL, and on the right side, the ratio of ESBL EC to E. coli. Of course, a typical outcome would be that upstream you have concentrations that are relatively low compared to the other concentrations. Then within the city, human waste often will have the highest ESBL E. coli concentrations, and animal waste often can have quite high concentrations. And downstream, in very many cases, we would expect to find an increase in concentrations as compared to the upstream sample. What we can see for this semi-hypothetical set, that if you look at the ratios, if you compare the ratio of ESBL in human waste, this is about 10% on average on a log scale. So what we see is that is compared to Dutch circumstances, in the Netherlands, we have about 1%, 0.1%. So that can indeed vary between countries. But we see also that the ratios can vary largely. So we need quite some sampling events to get to a number that you can use for statistical analysis, and that is intrinsic in the culturing methods, as we think. But I can conclude here is that the pressure in the human wastewater sample is probably larger than in the animal wastewater, because the ratio is lower in the animal wastewater, and that the downstream sample is probably mostly influenced by human waste, because the ratios are really quite similar there. So this is the kind of information we want to give the countries. Within the Tricycle protocol, there are also opportunities, as I mentioned, for extension of the protocol. So you can, if you want, detect residues of antimicrobials in different samples, for example, in water. We can use advanced epidemiological models. You can select additional sampling sites or additional populations if you wish to study exposed or risk populations. You can, of course, think about additional species. Within the JPI project Triumph, we work on methods for detection of carbapenemase producing Enterobacteriaceae that can be fed into the Tricycle approach if countries are interested. But there's many other species, of course, Salmonella, other pathogens. Of course, we stimulate usage of whole genome sequencing while acknowledging that it can't be done at all places in the world, but this should be used if possible. And we provide methods for analysis of those. And of course, we would like to link this with other surveillance systems and glass. Very shortly about the implementation, the protocol has been piloted in countries from different regions, importantly Madagascar, Ghana, Indonesia and Malaysia, and Pakistan were some of the countries that joined at the very first round. And there's more and more countries now implementing the Tricycle protocol. The protocol is indeed increasing, depending on financing through international agencies. What did we learn through piloting the system? Well, we indeed found out that implementing the Tricycle protocol can lead to a strengthened AMR and One Health advocacy. As I said, the protocol promotes integration across different sectors, and that is a very important outcome. And as mentioned by some countries, this can also help to overcome competing government priorities. There's competing scientific priorities, but also governmental priorities. And if you start out together, then it's much more likely that you work towards the same goal without competing. We noted that meticulous planning really helps, as in all projects, so that regards ethical approvals for sampling healthy women, memorandum of understandings are often needed, and also practically lab supply lists are helpful. Procurement can be terribly difficult, as we learned, even if you need antibiotic discs, and regular reviews of the approaches can also help. And in a general aspect that is often capacity building is needed. Trainings should also be repeated if possible online. Well, what are the next steps? The protocol is now published, and I note the only thing I forgot is to provide you with the link to the actual protocol in this presentation. So you owe me that, I owe you that. The protocol could be translated. We're working on online training courses to overcome the COVID travel restrictions. And at last, Tricycle module is established to help data collection. So what I would like to conclude, this is a personal view because there's many people involved in the setup of the Tricycle protocol. I think this protocol is feasible in various settings. It has moderate costs, it needs limited infrastructure. It's a public good, it's a published protocol. And I think this is also one of the very few examples of a truly multi-sectorial One Health project. I think even internationally, there's very few examples where the environment is integrated with the animal and human sector. So I think that's a great good. And of course, we recognize ESBL E. coli are just such a minor part of AMR. Of course, sampling 2 cities is not even nationally representative. Of course, this can be improved and amended according to the needs of the participating countries. So what we really hope are initiatives and enlargement. So we really hope this can act as a starting point, but we also should realize in many countries, even transportation of a sample for more than 10 hours is just impossible because we can't cool the samples or because transportation takes so long. So we just have to be aware that we have to distinguish the goal of a surveillance protocol, and this goal is to provide a protocol that is able to be achieved in most of the countries we have at hand. So the choice here is really simplicity with the hope for more. Yeah, that brings me to the end. I would like to thank many people, most importantly the ones from WHO headquarters and the ones we established the protocol with. And this work has been conducted within the WHO Collaborating Center for Risk Assessment of Pathogens in Food and Water at the RIVM. And also thanks to the funders of the JPI Triumph project, in which we try to extend the protocol to CPE and provide tools for genome sequencing analysis of isolates. So looking forward to your questions, but first I understand that Etienne is going to enlighten us. Thank you.
Thank you very much, Heike. That was a great presentation. And as Heike indicated, we want your questions, but please put them in the Q&A box that you should find at the bottom of the Zoom window. And then we will go through them after Etienne's presentation. So our next speaker is Etienne Ruppé. He is associate professor at the University of Paris and the Paris University Hospitals. His current work spans different aspects of antibiotic resistance, like carriage, diagnosis, infections, emergence, epidemiology, prevention, and also public information. And Etienne is also one of the 6 PIs involved in the EMBARK project. So the floor is yours, Etienne. Please introduce us to how we work with Tricycle.
Thank you, Johan, and thank you, Heike. So that I tried during your presentations to cut all the overlapping parts, but I still kept some of them because I think I want to highlight some aspects of Tricycle that connect to the EMBARK project. So these presentations are called EMBARK on the Tricycle. So there's going to be lots of words with the games with the words. So I'm sorry if this is not funny at all. So yeah, actually, Tricycle is something quite recent. Even if some countries have started to implement it a few years ago, actually it was officially released in this March. So with the official protocol. And I just want to remind the main objectives of Tricycle and just insist on a few notions. The main objective is to design and implement a multi-level, transsectoral, and multinational surveillance program for AMR. And actually, that's something that relates also to EMBARK. The surveillance, even if EMBARK is not going to be transsectoral, we saw that Tricycle it actually addresses environmental samples. Tricycle aims also to quantify and to track the status and dynamics of AMR among ESBL E. coli as a proxy indicator, and that's also something that makes sense in EMBARK. Of course, the aim of Tricycle is to be implemented in as many countries as possible from all the WHO regions. And I think most crucially, because this is, I think, the objective of any program for AMR is to, surveillance of AMR is to follow the impact and effectiveness of an intervention. We definitely need indicators to see whether any intervention is effective or not. So now, what are our EMBARK objectives? And there are two main objectives. First one is to establish a baseline for how common the resistance is in the environment, and let's say what type of resistance can be expected and where they can be expected. And the second objective, you see, is kind of close to Tricycle in a way. The objective is to standardize different methods for resistance surveillance that will be culture-based, but also molecular-based. And with the objective also to identify high priority targets. Like we saw that in Tricycle, we focus on ESBL E. coli. In EMBARK, we aim at identifying some maybe other ESBL E. coli, some kind of other targets that could be used for efficient monitoring. So at the end, EMBARK will produce a new monitoring scheme that will be designed so that it can be used modularly, depending on the available resources. So you can see how it connects definitely to Tricycle. So this is the organization of Tricycle. And as Heike showed you, we in EMBARK could be located here in this work packet 3, surveillance in environment, and a little bit here in the molecular biology, but you can see how EMBARK can be and so on integrated in this Tricycle organization. I also borrowed this figure to the Tricycle protocol, and so Heike detailed the kind of samples that are advocated in Tricycle. So to sample this type of environmental samples, the river upstream, a city, ideally the capital of the country, and let's say another a sentinel city, like a big one, not too far from the capital. And the scheme is similar to get some samples in a river upstream, the presence of humans, then to sample in a place where the wastewater meets the river, then also where some wastewater from wet markets meet the river, and then downstream the river. So you also saw that the number of samples was not that much, and In EMBARK, we had a broad imagination regarding the types of samples we could use, but definitely this type of sample would be integrated. So Heike mentioned also the list of countries that are already implementing Tricycle are here, and I put Pakistan in bold and in red because it's also a country participating to EMBARK, and these are the countries here that are, I'm going to show you here. No, that's the arrow. I don't have the laser here. So these are the countries in the process of implementation. And you can note that most of those countries are low and middle income countries. And this is the consortium of EMBARK with environmental samples taken from Sweden, Germany, France, Pakistan. So we have a proxy with Tricycle here, and China, but definitely we're mostly a Western Europe-based consortium. Now, Tricycle is based on culture, and culturing samples from the three sectors: human feces, poultry sector, water samples. And we in EMBARK are focusing on environmental samples, so water samples, but also soils, surface, like for instance, house gas structures, surface swabs, and also meat. And back to Tricycle, the media use are a McConkey for feces and Seca, and as Heike said, TBX media for water samples. And back to EMBARK, we use McConkey and pretty much for everything. So, but let's keep in mind what Heike said, that for water, we may need something else. And also look at the concentrations here. Tricycle is aiming at recovering ESBL E. coli, so they use a 4 microgram per milliliter concentration of cefotaxime. And we in EMBARK, as per the protocol, but that's, let's say, early stages protocol, we use 1 microgram per milliliter cefotaxime, so maybe more sensitive, maybe less specific. We can also note that we use other media supplemented with other antibiotics, penicillin, meropenem, ciprofloxacin, because we also aim at broadening the spectrum of bacteria we aim at recovering, so not only ESBL producing E. coli, but also parabepenemases producing bacteria such as XF48 producing enterobacteriaceae, and this media with temoxylin, but also like conventional other carabepenemases producers, just NDM type producers with this media with meropenem, but also bacteria resistance to ciprofloxacin. We also aim at monitoring concentrations of gram-positive with this mannitol salts agar and bile-esculin agar. And another difference is that in Tricycle, there's a filtration for surface water, and in the early stages protocol for EMBARK, we do direct plating for water samples. So you can see some differences here. So now, how can Tricycle embark on EMBARK in order to triumph? So that's the game. So that's kind of thoughts that we can discuss about. So the first thing is we just saw what we need to, at best, harmonize the culture protocols. They are not so different, but they have some differences. And I think Tricycle is like the core, like Heike showed in the last figure. And we can provide other culture protocols like for carbapenemases or other resistances, but I think that the core of the protocol should stick to what Tricycle is proposing. Also, I think we should at best try to harmonize the location of the chosen site where we're going to sample environmental samples in order to avoid the multiplications of surveillance protocols. And the second point is, so in EMBARK, we will propose, in addition to culture methods, some molecular methods. That's it's basically most of the work that's going to be conducted in EMBARK and many qPCR and metagenomics, and they could be considered as some optional bricks to recycle in a, let's say, a la carte fashion. Also, I think EMBARK is ideally would identify some other ESBL E. coli as potential markers. So Heike said that, of course, ESBL E. coli is not a reflection of all the EMR in one given environment, but this is a very nice proxy because it's pretty much present everywhere, but at different prevalences. So, and it's kind of dynamic, so it's a good marker in this perspective. So maybe from qPCR and metagenomics, we're going to find other proxies that could be used in complement to ESBL E. coli. And I will mention it at the end of my talk, the importance of networks and communication. So we had a NetES, GPR AMR funding network, and I will talk about that in the next slides. And also there's another network, which is called ShareNet. For the PI is Christian Lénard in France, which is taking over. So this is crucial if you want to implement some even very easy protocols such as, I mean, so-called very easy protocols such as Tricycle. It's not that easy. There's many hurdles that you have to discuss about. And in this perspective, we created a network that includes some countries which had implemented Tricycle and countries about to implement it, and also European countries that were involved in setting up Tricycle. And we had this conference in Paris in 2019, some teleconferences, and of course, because of COVID, we have to postpone the meeting in Brussels, and we have communication throughout the year through the WhatsApp group. So I like to see pictures where people didn't wear masks. So that happened 2 years ago. So you meet Heike here is just in the middle of the audience. And then we went to restaurant, but at the time it was open, but also we were studious and we did some very good work. So that's, I think that's very important. And if we want at some point to include some EMBARK stuff to Tricycle, then it will also require a lot of communication. So that's my last slide. I think the main perspectives of this protocols, Tricycle, EMBARK, and also Triumph, you didn't mention it, but Triumph is also some kind of add-on that we can add to Tricycle. And I think we want all to move towards standardization in order to make comparisons between countries, but also temporal dynamics in order to assess efficacy of interventions. And that was my last slide, and I'd be happy to take questions.
So let's see. We are getting a few questions. If I could ask you, Heike, to also unmute yourself.
There I
am. Let's start with some questions that I've been thinking about just in general here. Remember that you can post your questions in the Q&A. So one thing that has stricken me in general here when it comes to the Tricycle protocol is that there is, as you say, as both of you say, one of the purposes is simplicity. But that also means that you get some issues with representativeness. And I wonder if you could comment on that. How representative is E. coli as a representative, or ESBL E. coli as a representative of resistance in total?
If I may start, it depends very much on what kind of resistance you're interested in, because I think what we can distinguish is forms of resistance that are brought about mainly through selective pressures acting in animals and humans that are then through fecal matter introduced into the environment and then might lead to human exposure through the environment. And for those, I would argue ESBL E. coli is quite a good marker, because we can culture it, it is present, we also know it's relevant, and it's selected for in animals and humans. So there, and E. coli is used as a fecal marker widely. So you could argue that you could also look at enterococci. Yes, you can also look at enterococci if you wish. But for this form of resistance, which is mainly moving through the environment from humans or animals to humans, I think this is a good proxy. But of course, it's a very, very bad proxy for resistance that actually stems from the environment, and meaning that genes that are located on environmental bacteria, maybe not even in a mobile form, can be mobilized into new genetic elements. Mobile genetic elements can then be mobilized into different species, and finally can be mobilized into clinically relevant species. And for these type of genes that could potentially be mobilized, ESBL is no marker at all. So yes, and for some issues it is representative, for some issues it's clearly not.
Etienne, do you have anything to add to that?
Yeah, I can comment. E. coli is also a wonderful proxy because it's present in humans, in all humans, and all mammals. So that's it's present in the two sectors, animal and humans. And also, I think the choice of ESBL E. coli was driven by the fact that ESBL E. coli was the first pandemic of a community multi-drug resistant bacteria. It started at the end of the 90s, and then it just spread in many countries, especially in tropical regions. And that was the beginning of the pandemic of this multi-drug resistant bacteria, and then it was followed, for instance, with a second step, we would say, with the carbapentamases producers, such as NDM and oxafolate, that are also spreading in E. coli in the community. So from the clinical perspective, choosing E. coli with ESBL it really makes sense, because that was the main threat in the 2000, and now maybe we could move to the carbapentamases in some settings, because I think in some countries, ESBL E. coli is not anymore an issue. It's so much present. It's not a good marker anymore because it's so much prevalent. So we could move to complementary markets.
Any final comment on that, Heike?
No, sorry, because I was answering a question. Sorry. But I perfectly agree with the choice for E. coli as being a intestinal organism that is present in many species. Yes.
Yes. So that we have a bunch of questions from the audience as well. One of these questions I think you have already answered, Heike, but I think we were bringing that up anyway. So could one use coastal waters to represent the environmental arm in tricycle studies?
Yes, if that is accepted as an add-on to Tricycle, and if that is relevant for that particular country, of course, because many countries, or that will differ largely across countries, how important coastal water is for human exposure, of course. So, yes, yeah.
So following up on that a little bit, I lost my thought there, so whatever. Let's move to more audience questions instead. Is it possible to start collaborating with Tricycle or EMBARK? There's an example here of an Erasmus plus capacity building project based on AMR in Benin and Burkina Faso. What do you say? How easy is it to collaborate with Tricycle?
For Tricycle, what I would suggest is, up to now, Tricycle has been run predominantly by governmental agencies, and this most often followed the route through the national WHO office. And what we saw is that WHO has a critical role in engaging actors on a national scale, finding the correct actors, and also ensuring that this will have a sustainable impact. Because if it's directed at research institutions, it might be more difficult to implement surveillance on a longer time scale, because it becomes less of a research question. So what I would encourage is, yes, of course, we would like to have as many countries participating as possible. And I personally would also encourage you to get into contact with the local WHO representatives to discuss possibilities.
And connected to that, if a country is not in the list of participating countries, what would be needed to be included in Tricycle?
I think what's needed is to send Jorge Mateo a message at headquarters and discuss that with him, because what is needed is the most important thing is for Tricycle, from my personal viewpoint, is What we would really like to encourage is an intersectoral collaboration. So we're not so much into some environmental agencies starting out with environmental testing, but I think it makes most sense if you already have, or if you're aiming for collaboration between the animal, human, and environmental sector. So what's needed is the willingness to do that. And yeah.
And I think also what's needed is money. And you mentioned the sustainability, and many countries have started to implement Tricycle and have succeeded in securing the money for 1 or 2 years. But clearly the challenge is how to move forward and how to get the money for further years and to have something, you know, sustainable. That's one of the biggest challenges, because I think, maybe correct me if I'm wrong, Heike, but I think WHO is it's not directly funded, or maybe in some exceptions, some Tricycle projects, but most of it is funded by other sources, which are not, I mean, guaranteed every year. So it's just another competition, and it's taking time and energy. So that's clearly a challenge.
That is true. So for many countries, or for countries which are involved in Fleming Fund country grants, these support Tricycle approaches as well. So if you are from a country where Fleming Fund supports your AMR surveillance, you can clearly call for Tricycle approaches, because that's supported. If that's not the case, then you have to provide your own finances indeed. And for sure, there's so far a lack for example, European countries to engage in Tricycle. Not even we in the Netherlands have formally conducted Tricycle. So that would be great if there would be some countries to also have some comparative assessments.
Yes. So I think maybe with that we can step back a little bit and look a little bit more at the major objectives of monitoring. So I have a question from Joachim Larsen here. It's addressed to Heike, but I think it's actually something that we both could have something to say about. What is the ultimate objective of environmental or sewage sampling of ESBL in Tricycle? Is it better to understand transmission risks of ESBL via the aquatic environment back to humans, or do you have other objectives in mind?
This is, of course, a very good question. I think, to my point of view, one of the questions is indeed the transmission back to humans, therefore the inclusion of the river as a very bad and circumstantial starting point for one medium where you would probably have some human exposure. And of course, if you truly want to study transmission through the environment, you would have to enlarge that, but this will help to trigger awareness of the presence of ESBL, for example, in surface waters, because it's surprising how little data there is on ESBL in surface waters globally and how that might lead to human exposure. So that is one goal of those. Also, ideally, from the comparison of the ESBL isolates obtained from the environment and from humans and animals, one in the end could also conduct molecular similarity analysis and therefore gain an impression of how similar, for example, the animal isolates are to human isolates, and whether some isolates from water are more similar, which is very likely, as they are most likely influenced by human-based fecal material as well. It also points to the sources. So you could also argue, if you look at least the water compartment, what we often find most likely is that human fecal matter is more important than animal fecal matter for contamination of a city river. So that might also give you some glimpse of the relevance of sanitation measures that are directed to human discharges. And most importantly, probably we can't answer this with the Tricycle methodology properly, but what we can is provide data to make a starting point there and show what's needed to really answer those questions.
So I'm going to let Etienne answer this question as well, if he wants to. But first, it sounds that this is still mostly about understanding transmission risks then. Yeah.
Yeah,
I'm getting
together with the world of the
kind of
sources. Yeah, so look at sources of animal and humans versus each other, and then looking at transmission risks. Yes. Yeah.
So Etienne, do you have anything to add to that question?
Well, I don't have much to add, and I would just say that, of course, the isolates of E. coli that can be identified in the environment can be compared to the other sectors, and of course, the E. coli causing infections, because we have the E. coli causing bloodstream infections in the hospital. And this is, of course, answering one question is, are those E. coli causing infections, how are they close to the E. coli we can find in environment, for instance? So is it really a risk? I mean, it's just like colonization, or at the end, can they be involved in infections?
Thank you. So another question of sort of the expansion possibility of Tricycle. Are you checking for fecal materials of poultry only as an additional step? Are there plans to expand this to ESBL E. coli in other livestock like cattle, sheep, and goats? Is another audience question here.
That's for the first time I heard this question, actually. We have never considered animals other than poultry, actually, for the reasons named. But again, if a country thinks that sheep is predominantly eaten, then it makes perfect sense to also look at sheep as an extension. Yeah.
And I guess that would be one of these sort of extensions or add-ons that you talked about.
Exactly. Yeah. Yeah.
So a follow-up question on that: how are these add-ons integrated into the broader Tricycle protocol? Is there some systematic way of doing this, or is it more ad hoc?
So far, we had some add-ons for which we have a method specified in the protocol already. We haven't really discussed how to integrate new add-ons that will evolve. And I'm speaking on behalf of WHO here. I have no idea, but I think one procedure to bring that about is provide regular updates depending on what countries come up with additional needs and what kind of protocols can be developed. And some of those would be more straightforward, and some of those would require more resources, I think.
Etienne, any additional thoughts there?
Yeah, the comment is that it's really the beginning of Tricycle. I mean, the protocol was only released officially just 2 months ago, and I think, as Heike said, it's very open. I mean, you have this core Tricycle. I think you have to stick in order to enable comparisons. But I think then you can make whatever makes sense. But ideally, all those add-on should also be harmonized and discussed thoroughly. So that's of course you have the trial project, which will be very useful for the carbapentamases add-on. And I think EMBARK is typically the kind of place where we can discuss the possible add-on based on molecular methods.
I was actually thinking about this myself, but it feels like these add-ons are pretty much the kind of structure that we want to achieve in the EMBARK program. We want to be able to add this information and have it in a standardized way. So I think that this is one of the places where EMBARK could contribute, at least for environmental add-ons. I don't know so much about clinical add-ons. So thank you very much to both of you for participating today. We are approaching 3 o'clock in Sweden, so the webinar is approaching its end. It was two very interesting talks, and very nice to hear so concrete examples of how to implement these protocols in different settings. And if there are any further questions, should people address them directly to you, or how do you want questions?
Yeah, of course.
Yeah. So please send questions to Heike and Etienne if you have further questions that we couldn't take up today due to time limits. I would like to just say a few final words on the upcoming webinars. So the next one is in 3 weeks, and we will be a presentation by Dr. Maurice and what lurks behind the role of water in the transmission and persistence of AMR. And then the last webinar for the spring will be on the last of June. It will be a presentation by Kimberly Klein on Enterococcus faecalis biofilm-associated infections. And then we have a few webinars for the fall already booked. So starting the 1st of September, Amy Pruden will present on coordinating surveillance of resistance in wastewater treatment plants. Gautam Dantas will be talking to us about combating perturbations in microbiomes. And Barry McMahon will talk a little bit about how wildlife influences AMR. And there will also be at least 3 more talks before Christmas there that will be announced on the website soon. So thanks a lot for all of you who attended today. Thanks to the 2 speakers, and I hope to see you again in 3 weeks.
Thank you, Johan. Thank you, Heike.
Thank you.
Bye.
Nice initiative.
Bye-bye.